Prescription drugs that inhibit enzyme activity. Several common types of prescription drugs have modes of action that involve enzyme inhibition. Included among them are ACE inhibitors, sulfa drugs, and penicillins. ACE inhibitors are used to treat high blood pressure conditions as well as several heart conditions. Sulfa drugs and penicillins are two well-known families of antibiotics. Discussion of how these three types of medication function in the human body in terms of enzyme effects is illustrative of how enzyme inhibition can be used in a positive manner in treating human diseases. The acronym ACE stands for angiotensin converting enzyme. Angiotensin is an octapeptide hormone involved in blood pressure regulation. It increases blood pressure by narrowing blood vessels. Until needed, angiotensin is present in the body in an inactive form as zymogen. So you call this angiotensinogen, which is a decapeptide, meaning there are 10 amino acids. ACE converts the inactive decapeptide zymogen to the active octapeptide form, angiotensin, by cleaving two amino acids from the zymogen structure. So this portion here is removed to become angiotensin. ACE inhibitor medications block the action of ACE in converting angiotensinogen to angiotensin. The effect of this is a lower blood pressure than if the zymogen activation had occurred. Representative of ACE inhibitors in use today is the compound lisinopril, a compound heavily prescribed for treatment of moderately elevated blood pressure conditions. Structurally, lisinopril is a 1-carboxy 3-phenyl propyl derivative of the dipeptide lysylproline. In 1932, discovery of the antibacterial activity of sulfanilamide led to the characterization of a whole family of sulfanilamide derivatives, collectively called sulfa drugs, the first antibiotics in the medical field. An antibiotic is a substance that kills bacteria or inhibit growth. Antibiotics exert their action selectively on bacteria and do not affect the normal metabolism of the host organism. Antibiotics usually inhibit specific enzymes essential to the life processes of the bacteria. Sulfanilamide inhibits bacterial growth because it is structurally similar to PABA or para-amino benzoic acid. Many bacteria need PABA in order to produce an important coenzyme, folic acid. Sulfanilamide acts as competitive inhibitor to enzymes in the biosynthetic pathway for converting PABA into folic acid in these bacteria. Folic acid deficiency retards growth of the bacteria and can eventually kill them. Sulfa drugs selectively inhibit only bacteria metabolism and growth because humans absorb folic acid from their diet and thus do not use PABA for its synthesis. Shown in this figure are the most common sulfa drugs and their structure. So the general structure is this. Okay. Take note of the R group. If the R group consists only of hydrogen, then you call it sulfanilamide. If it is another group like this one, a carbonyl group, you call it sulfacetamide. Okay, and so and so also you have sulfisoxazole, sulfadiazine, sulfadimethoxine. So this is arranged according to their increasing effectiveness. So this is the most effective and this is the least effective. Penicillin, one of the most widely used antibiotics, was accidentally discovered in 1928. A decade later, scientists were able to isolate penicillin in pure form and proved its effectiveness as an antibiotic. Several naturally occurring penicillins have now been isolated, and numerous derivatives of these substances have been synthetically produced. All have structures containing a four-membered beta-lactam ring fused with a five-membered thiazolidine ring. As with sulfa drugs, derivatives of the basic structure differ from each other in the identity of a particular R group, this R group. So if this R group consists of a benzyl group, it is penicillin G. If the R group consists of this, you call it penicillin V. And so you have other derivatives such as methicillin, ampicillin, amoxicillin, oxacillin. Penicillins inhibit transpeptidase, an enzyme that catalyzes the formation of peptide cross-links between polysaccharide strands in bacterial cell walls. These cross-links strengthen cell walls. A strong cell wall is necessary to protect the bacterium from lysis, or the breaking of the cell. By inhibiting transpeptidase, penicillin prevents the formation of a strong cell wall. Any osmotic or mechanical shock then causes lysis and killing the bacterium. This slide illustrates the selective binding of penicillin to the active site of transpeptidase. So this is the enzyme. This portion here is the active site, and it contains a serine residue, and this is penicillin. Penicillin forms a covalent bond. So you have here the covalent bond with the serine residue, resulting in irreversible inhibition of the enzyme. So the penicillin permanently blocks the active site. Enzymes can be used to diagnose certain diseases. Although blood serum contains many enzymes, some enzymes are not normally found in the blood, but are produced only inside cells of certain organs and tissues. The appearance of these enzymes in the blood often indicates that there is tissue damage in an organ and that cellular contents are spilling out or leaking into the bloodstream. Assays of abnormal enzyme activity in blood serum can be used to diagnose many disease states, some of which are listed in this table. So you have here the presence of lactate dehydrogenase. This is indicative of heart disease, liver disease. If you have creatine phosphokinase, indicative of heart disease. Okay, and so on. You have aspartate transaminase, alanine transaminase, gamma glutamyl transpeptidase, alkaline phosphatase. Okay, so these are the indications that the disease is being indicated by the presence of those enzymes. Enzymes can also be used in the treatment of diseases. A recent advance in treating heart attacks is the use of tissue plasminogen activator, which activates the enzyme plasminogen. When so activated, this enzyme dissolves blood clots in the heart and often provides immediate relief. Grapefruit effect. Grapefruit and grapefruit juice also contain compounds that can slow down the metabolism of several common prescription medications by interacting with the enzymes needed for their metabolism. Example is liver enzymes called cytochrome P450S. This enzyme inhibition, known as the grapefruit effect, leads to increased concentration of the affected drugs in the bloodstream, which increases the risk of potentially serious side effects from the drugs. More than 50 medications of diverse types are now known to be affected by the grapefruit effect. A partial listing of affected medication is as follows. So in this slide, you are given a partial list. Okay, so examples of drugs affected by the grapefruit effect, you have amiodarone, buspirone, carbamazepine, etc. This grapefruit effect is significant enough that the United States Food and Drug Administration, FDA, now requires that all new medications be tested for problems arising from grapefruit interactions before the medication is approved for use. Drugs whose metabolism is affected by grapefruit and grapefruit juice must carry warnings in their patient information sheets. The actual compound responsible for the grapefruit effect have been identified. They are the compound bergamotine and one of its metabolites, dihydroxy derivative of bergamotine. Both compounds have a fused three-membered ring structure containing ether and ester functional groups to which an unsaturated carbon chain is attached.